My name is Zachary Zimmerman, and I'm a biology teacher for Dysart Unified School District in Arizona. And I just want to share with you how I teach photosynthesis to my AP Biology students. And I've shared with you a link to a couple of documents. First, my lesson plan, and then also a personal essay on why and how I teach the way I do. But probably the most foundational aspect of being a great teacher, which I always aspire to be, and I work every day as hard as I can at the skill. That foundation has to be the relationships that we build with our students. And so that relationship is built on trust, mutual respect. And so by the time I teach photosynthesis to my students, they and I will have had several months to develop that working relationship. And so I encourage my students to take risks, to ask questions, even if they may end up being wrong when they try to answer those questions, whether it's a scientific question on photosynthesis or anything else. I teach them that that's always a learning experience. So when their hypothesis ends up being rejected, that's okay, because we've learned something from that. And so once my students feel that they can take risks and use their errors as opportunities for learning, then it makes for a very relaxed and productive learning environment in my lab. So now let's get to the photosynthesis lab. These AP Biology students, many of them are training to be scientists in the future, whether they be doctors, health professionals, or maybe some of them might go into engineering, but they are training to be scientists. And so I try to help them think scientifically. They need to be problem solvers. They need to take a critical problem and try to find a solution that's, if not novel, maybe has a unique characteristic. And so I invite them to take those risks, and they're going to, of course, go through the scientific process. So they first ask a question: what is it they want to know? And then secondly, once they formulate that question, they're going to state a hypothesis. And then the students will take that hypothesis and use it as groundwork for the building of their experiment. They actually design their experiment, and they will implement all the right controls so that they can trust the results that they get. They will also, of course, have a way of generating quantifiable data, or quantitative data, I should say, and then they're going to graph their results. And my goal for them is that they be able to explain in writing and in spoken word their results. They should be able to know whether their hypothesis was rejected or accepted. They should be able to tell me what the significance of the results are and explain to me, as if I have no idea what the data means, what those graphs are actually telling us. And so I have them go through the scientific process in every single unit that we go through. Real-world applicability is so important. So before the lab, during the lab, and even during discussions near the end of the lab or at the end of the lesson, I invite students to think about the real-world importance of what they're learning. There are a lot of agricultural applications that could be explored as a result of a photosynthesis lab. There are biotechnological applications as well. And when we read about the continuing climate change, if there's an increase in frequency of droughts in certain areas, well, how can farmers be more productive? How can they utilize resources? How can we be better stewards of resources such as light, or such as water, and nutrients and so on in the soil? And so students can explore what they're learning in the lab in light of these real-world problems. And so I wanted to also share with you the details of how the lab is carried out. So first, these students are going to take spinach leaves, and I have them use hole punchers. They punch out little discs from those leaves, and then they place those discs into a solution. And the solution has water in it, of course, but it also has some sodium bicarbonate. They use baking soda as a source for that. And so the baking soda, of course, provides the carbon dioxide. Students will take syringes with plungers. They'll take the plungers out and they'll put into the syringes some of the bicarbonate solution that they would have prepared earlier, and they'll also take the leaf discs that they would have punched out using hole punchers and place them into the syringe as well. And then they put the plunger back in, and then they basically create a vacuum. And the purpose of this is to take the oxygen that's inside the spinach leaves and remove that, basically squeeze or push that out. And then that way the leaf discs, once they're put back into the cup with the solution, will not stay afloat. They will drop because that decreases their buoyancy. And so the students have to make sure all the leaf discs that they use in their experiment has gone through this process. And so once they've done that, then they can explore whatever variable that they choose. They can choose an independent variable, such as the wattage. So I might provide for them a number of different bulbs of different wattages. How does light intensity affect photosynthesis? Another lab group may choose to investigate the concentration of sodium bicarbonate. I had another lab group that investigated how wavelength influences photosynthesis rate. Another lab group may investigate another independent factor, such as maybe the species of plant. Maybe instead of spinach, use some other kind of species. Another factor could be stomata density on the leaves. So whatever independent variable they choose, they commit to that, and then they will design their experiment around investigating the influence of that independent variable on the dependent variable, which would be photosynthesis rate. So students were instructed, of course, to assess photosynthesis rate as being indicated by the rise of those leaf discs in the solution. So going from the bottom of the cup to the surface. Now one might ask, well, how does this happen, or why does this happen? Well, the students infer that as the leaf discs rise to the surface, this is because of photosynthesis. And of course they reference their background learning, and they of course would have been exposed to the equation of photosynthesis by this point. They would have read quite a bit about it, but the products of photosynthesis are organic compounds and oxygen. And so as the oxygen is produced in the mesophyll, spongy mesophyll of the leaf, that increases the buoyancy of the leaf discs, and then the leaf discs, once they reach that critical threshold as far as buoyancy, they start to rise to the surface. And so the amount of time it takes for that leaf disc to rise is a measure of its photosynthetic rate. So my students will spend several days in the lab investigating the influence of their chosen independent variable on photosynthetic rate. And of course, they're going to be using my input and advice whenever they ask for it. I will give them whatever words of wisdom I can give them on how to best design their experiment. But I don't just spoon feed the game plan to them. I have them develop the game plan for themselves or amongst themselves, because it's a group of 3 or 4 in a lab group. And I encourage them to think of themselves as professional scientists. I mean, they are professional students after all, so they may as well think of themselves in a science class as being in a professional lab and producing a professional presentation. So that's one aspect of their learning or evidence of their learning. It'll be a slide presentation that they produce, and they'll talk to their classmates. Their classmates will ask questions at the conclusion of that 10-minute presentation. Each person in the group is expected to report on what they learned, different aspects of the experiment, whether it be introduction and hypothesis, or maybe another student will report on the data that was gathered and try to make sense of the graphing that the audience is maybe trying to understand. And then also another student may report on the results or maybe the conclusion part of that. So for the written report, they will generate a paper, a scientific paper, as a group. It'll be organized the same way as any scientific paper in any journal, and it will have an abstract at the beginning, although this is usually prepared last once the rest of the project is complete. The students will put an introduction in there. They'll explain the background information, they'll state their hypothesis, and then of course they devote at least a page or so to designing or talking about the design of their experiment. They'll talk about the different constants that they used to try to really isolate the influence of the independent variable. They'll also have a section on data and the results that they got, and they're going to have graphs, and I give them some pointers on how to graph, but I expect them to follow those pointers. I do give them resources and examples to follow, which you'll see in the links that I've provided. And of course, the last part of that paper that the group generates is the discussion, where they talk about not just their results, but why they think they got the results they did, and whether their hypothesis was supported or rejected, why was it supported, why was it not supported, and they basically refer to their evidence. And I tell them, don't make any categorical claims that your hypothesis is wrong or right. Simply state whether it was supported or whether it was rejected. And that way they're leaving the door of possibility open to some other lab group, in theory, that might investigate that same independent variable influence, and they may end up with a different result. Maybe they design their experiment a little differently than this original group did. So I try to encourage my students to use scientific terminology and to basically think like scientists and to solve problems like world-changing scientists do. I think one of the most exciting aspects of a lab activity such as this is that my students have an opportunity to become experts in their particular topic or in their scientific problem that they were trying to solve. And they may, by the end of the process, end up knowing far more about that topic than anyone else in the classroom, whether it be their peers or their own teacher, which makes it quite interesting, not just for students, but for myself as the instructor. And so my students feel like they have something meaningful to contribute to the learning experience in the class, and that makes it a whole lot more fun for everyone. So as you check out those links and you review those documents, the lesson plan, personal essay, I hope that you'll appreciate how much learning is going on in my classroom and how much my students enjoy becoming experts on their research topics.